Hi everyone, let's get started. Today we're going to be talking about microbial genetics and specifically about the process of DNA replication and the problem of mutations. Now before we get into some of the details of DNA replication, we need to go through some vocabulary to remind ourselves of the definitions of some very important terms. First, we need to define this term gene. A gene is a piece of DNA that contains two very different types of code in its bases. It contains the code for building a protein, and it also contains regulatory code. Code that will determine if that protein is going to get made, when it gets made, and how much of it is going to get made. Second, we need to define this term genome. The word genome refers to an organism's entire collection of genes, all of the genes that are inside the nucleoid in that microbial cell. Then there are two terms, genotype and phenotype, that we need to be familiar with. The term genotype refers to the collection of alleles in an organism. So it's easy to confuse genotype and genome. The genome is the collection of the actual genes that any organism has. So every organism has a genome. But each individual has a genotype. Remember that even though all human beings share the same set of genes, each one of us has a different combination of versions of those genes. Those versions are called alleles. So, for example, all humans have genes that are associated with determining the color of their hair. So, the genes for hair color are in everyone's genome. Your version of the hair color gene might be different from my version of the hair color gene. So your genotype might have the allele for blonde hair, and my genotype might have the allele for brown hair. We both have the hair color gene, but we have different alleles of it. That's why genotype is different from genome. Genome is about your collection of genes. Genotype is about your collection of specific alleles for those genes. Now the last term is phenotype, and this one can also be a little confusing. If you translate the word phenotype, it actually means show form. So a lot of people associate show form as what you look like, what you show. That's actually an incomplete definition for phenotype. Your phenotype is your collection of traits. It's the collection of traits that an organism has. Now, a trait is simply a characteristic. We have to remember, though, that you have visible characteristics and you also have invisible characteristics. So it's not just about your physical show form. It's also about your biochemical characteristics and your behavioral characteristics. All of your traits, the traits that you can see and the traits that you can't see, are part of your phenotype. Now, in order for us to talk about DNA replication, we have to remember a couple of critical components of how DNA functions in a cell. Remember the central dogma. This describes how information flows in a cell, or what we often refer to as gene expression. The dogma states that DNA is used to make RNA through a process called transcription. RNA is used to make protein through a process called translation, and it's proteins that give us our traits. We've said a couple of times now throughout the semester that all cells use DNA as genetic material. It's important for us to remember that. It's also important to remember that the code that's in that DNA is the same for all living organisms. All cells use the same code to build proteins. And that code is actually in the form of 3 bases together, called a codon, that code for one amino acid. So the code inside DNA is based on groups of 3 bases. And again, we refer to those groups of bases as codons. And each codon codes for an amino acid. All cells use the exact same genetic code. All microbial cells do it, and all higher order organisms do it as well. So let's take a look at the drawing on this slide. This is a slide that walks through the process of gene expression visually, and it helps us to recall the complementary nature of nucleic acid. So if we start up at the top, you can see a piece of DNA drawn in blue, and the double strands are wound together on either end of this structure. And right here in the middle, the 2 strands have been separated apart so we can see the complementary base pairing. Now, we have to imagine that this is a gene. This is a little piece of DNA inside the organism that codes for a protein. And it also has regulatory code inside of it, code that regulates when that protein gets made. So here is the sequence, let's imagine, of the protein coding portion of the gene, 2 strands. Now, in the next step, they have separated those 2 strands away from each other. And what's happening is we actually see that one strand is being used as a template in order to build this red strand, which is not DNA, it's RNA. So when we talk about gene expression, we have to remember that the actual code for building the protein is found in only one of the 2 strands of DNA. And we call that one strand the template strand. So the cell will use this one strand, the bottom one in the drawing, to make an RNA off of. And that's what's happening here. And notice that between the DNA template strand and the RNA, we have complementary binding. The only difference is that there are no T's in RNA, only U's, uracil. So where we would expect to find a T, in RNA it substitutes with a U. So for example, if there's a T in the DNA, there'll be an A in the RNA. That makes sense? When there's an A in the DNA, however, there are no T's in RNA, so we have to use a U there for uracil. If there's a C in the DNA, there'll be a G in the RNA. And over here you can see if there's a G in the DNA, there'll be a C in the RNA. Complementary binding. Now, an RNA will be made across the whole protein coding portion of this gene. And then that RNA is going to be used to build the protein. The RNA will be used for the process of translation in order to build a protein. And what they're showing us in the picture is if we break the RNA into groups of 3 bases, each one of these groups, each one of these codons, codes for a different amino acid, shown here as these purple circles. So the ribosome, which does the work of translation, is going to read AUG as the first codon on the RNA, and it's going to say AUG is the code for methionine, and it will place a methionine in that first position. Then it will read AUC, and it will know that AUC is the code for isoleucine. That's this second purple circle. And it will join those together with a peptide bond because it's building a protein. The third codon, UCG, is the code for serine. And that amino acid will be added on to this growing strand. The ribosome reads the whole mRNA and builds this protein. And we can find the genetic code in any textbook, any biology textbook. You don't have to go to a microbiology textbook because we all use the exact same code. Remember what this genetic code table tells us. It is the table that shows every possible combination of 3 bases. So if we take the A and the U and the G and the C, and we put together all possible combinations of 3, we get this. All right? And the other thing the code shows us is, according to the codon, what amino acid it codes for. You can see that some amino acids actually are coded for by 4 different codons. And some amino acids are coded for by 2. There's even one amino acid, methionine, that's only got 1 codon for it. So this is the table that shows us which codons belong with which amino acids, the genetic code. So when it comes to gene expression, and remember, gene expression is described in the central dogma, prokaryotes are using their DNA to make proteins, just like eukaryotes are. Microbial cells are using their DNA to make protein, just like other cells are. But we have to remember that gene expression is not the only thing that cells use their DNA for. We have to remember DNA replication or DNA synthesis as well. DNA replication is the process where DNA is used to make more DNA, where the cell is going to make a complete and exact copy of its DNA molecule. Now I want to take a moment here before we get into the details of DNA replication to remind us that prokaryotic cells are capable of something very interesting that they can do with their DNA, which is transfer it horizontally. Remember, when we talk about vertical transmission of DNA, the vertical term means parent cell to daughter cell. So the same way that a human parent would pass its DNA down to its child, a parent microbial cell can pass its DNA to its daughter cell through vertical transmission. But microbes, prokaryotic microbes anyway, are also capable of horizontal transmission. They're capable of passing their DNA from their own selves to their neighbor. So they're capable of passing it to another cell of the same generation as their own. This is a very unusual process. It's something that you don't see outside of prokaryotic cells. I've listed here the 3 ways that horizontal transmission of DNA occurs. There's a process called conjugation, a process called transformation, and a process called transduction. Each one of these passes DNA horizontally, neighbor to neighbor, but it occurs using a different process. Conjugation is a process that we think of as being the sexual transfer of DNA from one prokaryotic cell to another. Transformation is a process where one prokaryotic cell releases its DNA into the environment, and then another prokaryotic cell takes it in. And finally, transduction is a process that occurs with a viral middleman. So there are viruses called bacteriophages that infect bacterial cells. And what happens is it's the virus that helps transmit DNA from one bacterial cell to another bacterial cell. So what we have on this slide is a drawing depicting the two kinds of ways that prokaryotes transmit their DNA. Vertically, which is what we think of as the classical way that you transmit your DNA from one generation to the next. And that's what they're showing us on the left. Here's a parent cell, and you can see the nucleoid, the circular chromosome in the cytoplasm. Now the parent cell is elongating, it's going through binary fission. And during that process, this DNA molecule has to be replicated, it has to be copied. That elongating parent cell then divides and produces two daughter cells that each have a copy of the DNA molecule. This is classic vertical transmission from parent to offspring, from one generation to the next. Horizontal transmission doesn't work like that. Horizontal transmission moves DNA between two cells of the same generation, two unrelated cells in the same generation. And remember, we said it can happen three ways. Conjugation is actually shown down at the bottom here. Conjugation involves a structure called a sex pilus. Remember, we talked about pili. We talked about these hair-like structures that we sometimes find projecting off of the outside of prokaryotic cells. Well, there's a type of pilus called a sex pilus, which can connect to bacterial cells. And we call it a sex pilus because it is essentially going to transmit material from cell to cell through this tunnel, through this channel between the two cells. And that's what they're showing us in this diagram. One cell is moving DNA, oftentimes plasmid DNA, from here across the pilus into the neighboring cell. The second kind in the drawing is this process of transformation. This one's a little strange to imagine, but in transformation, one cell is actually dying. One cell is going to die and fall apart. And in the process of falling apart, its DNA gets released. That's what they're showing us here. When DNA gets released out into the environment like this, upon the death of a cell, it's often broken up, it's often fragmented. Where it becomes horizontal transfer is when another cell, a living cell, takes in those pieces of DNA and incorporates them into its own cell. And last, we have this very interesting process called transduction. In this process, a virus is involved. They've drawn the virus here, and we haven't talked about viruses in a whole lot of detail, but remember that in general, the viruses inject their DNA into the host cell, and then the host cell replicates the viral DNA and then packages it back up and makes new viral particles that are then sent out of the cell. Well, during this process, sometimes mistakes get made. Sometimes, in addition to packaging up viral DNA, the cell also packages a little of its own DNA into the virus. And what happens then is that the new viral particles, as they're exiting the cell, are actually carrying not only viral DNA, but also a little bit of the host cell's DNA. And then they're going to go and infect another cell. And when they inject their DNA into that new host, they're also injecting a little bit of bacterial DNA. I know it sounds confusing, but it is a method for a virus to transmit bacterial DNA between bacterial cells.